Welcome to the deep dive. Our mission here, well, it's pretty straightforward. We take all that detailed research you send our way, and we boil it down into the essential knowledge you need, fast.
And today we're diving into a really critical question. How do we actually measure life way down in the deep ocean? We're looking at a baseline inventory of deep sea fish, but really through the lens of the tools we use.
Yeah, the specific paper guiding us today is led by Iguchi and colleagues. It's called Utilizing Environmental DNA and Imaging to Study the Deep Sea Fish Community of Takuyo-Daigo Seamount. So the core mission for you, listening in, is to get why just using one survey technique down there is, well, it's basically obsolete now.
Right, and how combining these newer methods gives us a much, much more complete picture of life in these remote spots.
And the remote spot is the Takuyo-Daigo Seamount. It's deep in the northwestern Pacific.
Exactly, and it's not just any underwater mountain. It's kind of a perfect natural lab for this kind of comparison, but also, quite fragile.
Okay, so to really get why this research matters so much right now, we have to talk context. And that means deep sea mining.
Absolutely. There's this rapidly growing global interest in mining the deep seabed. They're mainly after these cobalt-rich ferromanganese crusts.
Or where do you find those?
Well, they're spread across deep sea seamounts. Just like Takuyo-Daigo, these underwater mountains are, potentially huge mineral reserves.
That's the big conflict, isn't it? These same seamounts are incredibly important habitats. They're like oases down there, supporting unique deep-sea communities. Some species might only live there.
And they also boost productivity higher up, which supports major fisheries. Think tuna, mackerel out in the open ocean.
So it sounds like we're running up against a deadline. We desperately need good baseline data before any large-scale mining kicks off, for environmental protection, for actually understanding the potential impact.
Exactly. And the problem, as you probably know, is that surveying the deep sea is just incredibly hard. It's logistically complex, super expensive, and you just don't get many chances to sample.
Right. So necessity meets difficulty. And that's what's pushing the development of these new tech approaches. Which brings us to the actual toolkit the researchers used here.
Okay, so they zeroed in on the Seamounts Plateau, depths around 930 to 949 meters, and they compared 4 different methods, pitting sort of traditional observation against these newer molecular techniques.
Let's break those down. First up, imaging. Method 1 was kind of the standard, right? The ROV Transect, that's the remotely operated vehicle.
Yeah, the robot sub.
It drives around, follows survey lines. They did about 13.3 hours of this. It doesn't disturb things much, and it's great for seeing creatures just doing their thing on the seafloor, sponges, fish swimming by, that sort of stuff.
Then you compare that to method 2, also imaging, but different. The Adoko Mark I lander. This is basically a camera system they just drop, and it sits on the bottom. Static.
Okay, so one moves, one stays put. What's special about the lander?
Well, the key thing here was bait. They used Pacific sari, a type of fish in several of the drops, to deliberately attract animals.
Oh, okay. So you bait it to draw in the more mobile species, maybe the predators, get a better look at them.
Exactly. If the ROV gives you the wide-angle view, the baited lander is like setting up a camera trap to get close-ups of the animals that come investigate the food. It shows you a different piece of the puzzle.
Got it. Okay, so those are the cameras. Then we move to the molecular side, environmental DNA, eDNA. This is the genetic stuff organisms leave behind, right? Skin cells, waste.
Mucus, scales, all floating in the water. So method 3 was analyzing standard seawater samples. They filtered water between about 1 and 3 liters per site and pulled out the DNA.
And how do they know which DNA belongs to fish?
They use specific molecular tools called primers, in this case the MyFish primers. They leave them as search terms for fish DNA. They target a specific gene, the 12S RNA gene, amplify it, and then sequence it to see which fish species were present.
Okay, seawater filtering. What was method 4? This one sounded really clever.
Yeah, this was using sponge samples. Sponges are incredible natural filters. They pump thousands of liters of seawater through their bodies every day to feed.
So the idea is the sponge is already concentrating the eDNA from the water.
Precisely. The researchers collected tissue from 3 different types of sponges: Pharea, Pheronema, and Geodia. They used the sponges as sort of biological eDNA collectors.
That is smart. Saves filtering huge amounts of water yourself.
It's a neat idea. But the real question is, how did these 4 methods stack up against each other? And this is where things get really interesting.
Okay, let's get into the findings. So overall, they found 18 families of deep-sea fish, and the types of fish were generally what you'd expect for that part of the Pacific. No huge surprises there.
And this is the absolute key takeaway for you when they compared the lists from each method. Not one single fish family showed up on all 4 lists.
Wait, really? Not one.
Not one. The mix of fish detected by the cameras was significantly different from the mix detected by the eDNA.
Wow. Okay, that's actually pretty shocking. It's like looking at the same scene with 4 different types of sensors and getting 4 different reports.
Exactly. It means if you'd only used, say, the ROV, you would have completely missed major parts of the fish community.
So relying on just one method gives you a dangerously incomplete picture.
Fundamentally, it changes how we have to approach these surveys. Let's dig into why they were so different. What did the cameras see that the eDNA missed?
Yeah. What slipped through the genetic net?
It was the big guys. The chondrichthyes, sharks, and rays. Specifically families like Pseudotriachidae, the false cat sharks, and Summiosidae, the sleeper sharks.
Hold on. The eDNA missed the sharks. The technique that's supposed to pick up trace amounts of DNA missed some of the biggest animals down there. How?
It's a known issue, actually. The specific MyFish primers they use, those search terms, they just aren't very good at latching onto and amplifying the DNA of certain sharks and rays, certain elasmo branch.
Ah, so it's a limitation of that particular molecular tool.
Right. The tool itself had a built-in blind spot for some key predators. Future work needs broader primer sets to fix that.
Okay, so that's what imaging caught, but eDNA missed. What about the other way around? What did eDNA find that the cameras didn't see?
This was interesting, too. The eDNA picked up 5 whole families of fish that were completely absent from the camera footage.
5 families. What were they like?
Generally small fish, often less than 10 centimeters long. Things like Gonostomatidae, bristlemouths, Bathygatidae, rat tails, Melamphidae, big scales, Barbrucidae, red velvet whalefish, and Howlidae.
And why would the cameras miss those? Too small.
Probably a combination of things. They might be too small to reliably identify on camera. Maybe they live a bit higher off the seafloor than the cameras were focused, or perhaps they're sensitive to the noise and light from the ROV and just stayed away.
So the eDNA catches their genetic dust even if the fish itself avoids the lens.
Exactly. Different methods are biased towards different types of organisms and behaviors.
We also saw differences just within the imaging methods, didn't we, between the ROV and the lander?
Yes, definitely. The baited lander, the Adoko Mark I, was really good at drawing in those sharks we mentioned, the Pseudotriachidae and Somnioidea. Makes sense. Bait attracts predators.
Right. Big fish like free food.
But the moving ROV was better for spotting the less mobile species, the ones that tend to stay put. Like the Chinosidae, those are anglerfish, sometimes called sea toads.
Ah, the sit-and-wait predators.
Yeah.
The lander probably just didn't drop close enough or they weren't interested in the bait.
Likely both. Its view is narrow, and those fish aren't actively cruising around. So even with cameras, you need both mobile and static, baited and unbaited approaches, depending on what you hope to find.
Okay, and what about within the eDNA methods? Seawater versus the sponges. You said the results were contrasting.
They were. Statistically, the deep-sea fish composition wasn't significantly different between them, but what they detected overall was quite different in character.
How so?
The seawater samples were, let's say, cleaner, more focused. About 82% of the fish DNA they found belonged to actual deep-sea species.
Okay, so pretty targeted. And the sponges.
The sponges detected more fish species overall, which sounds good, but they had a massive contamination problem.
Contamination. From what?
From shallow water fish. Get this. About 69% of the fish DNA found in the sponges was from shallow species, like subtropical rabbitfish, Sigonidae, stuff that definitely doesn't live at 900 meters.
Whoa. So the sponge acted like, well, a sponge, and soaked up DNA from the entire trip up from the seafloor.
That's the strong suspicion. As they pulled the sponges up through kilometers of water, they likely filtered all that surface water eDNA, basically swamping the deep-sea signal they were trying to get.
Man, that really highlights a huge challenge in deep-sea sampling, just getting the sample back untainted.
It really does. It's a major logistical hurdle.
Okay, so let's pull back and think about the broader implications here. The big message seems crystal clear. Using both eDNA and imaging isn't just a good idea, it's essential if you want a reasonably complete picture of biodiversity.
Absolutely. Any survey using just one approach is going to be insufficient, potentially dangerously so if decisions are based on it.
And this feeds directly into the whole deep-sea mining regulation issue, doesn't it?
Straight into it. This combined approach is what's needed for proper environmental baseline studies and impact assessments. The kind of rigorous science regulators need before they make decisions about allowing mining. We can't afford to base policy on a checklist that's missing half the residents.
But the study also points out where things need to get better. That sponge contamination issue, for instance. Is there a fix for that?
There is, thankfully. The researchers suggest that future sponge sampling needs a better protocol. Basically, collect the sponge and immediately seal it in a container on the seafloor before bringing it up.
Ah, protected from the water column on the way back.
Exactly, prevent that surface water contact. They also noted limitations in sample size. They need bigger water samples, more sponge tissue, just more samples overall to make the statistics more robust.
Makes sense. More data is usually better. And what about the molecular tools themselves?
Yeah, improvements needed there too. To catch those sharks and rays consistently, they need to use a broader range of primers, not just mayfish. And they also bring up using environmental RNA, or eDNA.
eDNA. How is that different from eDNA?
Well, DNA can hang around in the environment for quite a while after an organism is gone. It's stable. RNA, on the other hand, degrades much faster.
Okay.
So if you detect the eRNA of a species, it's a much stronger indicator that the organism is alive and present right now, or was very, very recently. It helps distinguish the living community from just, old legacy DNA floating around.
I see. So it adds a layer of, is this signal current?
Precisely. It helps refine the picture of the active ecosystem.
So wrapping up, the real significance here seems to be getting this crucial baseline data, especially for this specific, poorly studied region, the Marcus Wake Seamounts and Ogasawara Islands area.
Absolutely. This provides vital, concrete information for conservation planning, right as the prospect of deep-sea mining tests looms closer in these areas. We now have a much better blueprint for how to do these surveys responsibly.
Which leads us to our final thought for you, the listener. We just heard how much critical information whole families of fish, big sharks, tiny species was missed by using just one or two methods at this one seamount. So think about all the other deep-sea regions out there, potentially even more diverse, that we've barely looked at. How much life might be completely unknown simply because our past survey techniques were inadequate.
And perhaps more urgently, how quickly do we need to deploy these improved combined methods to understand these ecosystems before irreversible decisions about resource extraction are made?